Hi, I'm Vaughn Cooper, and I'm here to present this poster on the WISP surface signaling system in Burkholderia, which we found recruits a parallel two-component system, which in turn co-regulates surface attachment by EPS and our Fimbri. And most of this work has been done by a former postdoc in my lab, Mhatre. I'm at the University of Pittsburgh in the Department of Microbiology and Molecular Genetics, and I'm the director of the Center for Evolutionary Biology and Medicine. A couple key points. Burkholderia is like Pseudomonas in that it has a WISP signaling system, WISP standing for wrinkly spreader, but it has a key exception. In Pseudomonas, WISP-E phosphorylates the diguanylate cyclase WISP-R, whereas in Burkholderia, WISP-E appears to phosphorylate not a diguanylate cyclase, but rather a hybrid histidine kinase that we call WISP-H. And so our big question was, how does WISP signaling work? What is being activated? To answer that question, we used an evolve and resequence approach where we took mutants, these RSCVs or Rugo small colony variants, and we isolated them under biofilm selection. And we in turn took those mutants, whose genetic basis we knew, and selected them for planktonic growth, which acted as a suppressor screen. And then we did that one more round to determine in turn how the suppressor might be resuppressed and drill down into that system. And we found that growth of WISP-A or E RSCV mutants in planktonic conditions selected smooth colonies. Many of them had mutations that eliminated loss of function in an undescribed previously two-component system that we call Rho-SR or regulator of wrinkly. So our big question is, how do WISP and Rho-SR together control biofilm phenotypes? First, we established that WISP mutations, the identical WISP mutations that increase cyclic diGMP in Pseudomonas do not, in fact, increase cyclic diGMP in Burkholderia. We then conducted RNA-seq of WISP mutants to find out what they were activating, and we found that they somehow were activating genes involved in the synthesis of the Burkholderia exopolysaccharide cluster, or BEP, as well as genes involved in fimbrial biosynthesis. We then selected WISP-A Rho-R double mutants under biofilm conditions, which created a new RSCV mutant, which had insertion sequences that hyperactivated another gene called BurA, which is a cyclic diGMP transcriptional regulator that binding transcriptional regulator that activates the BEP cluster. And so the insertion sequence inserted right here. To put these model and phenotypes together, we find that there is this elaborate signaling system, which is much like the key regulon involved in surface sensing in Burkholderia. What that winds up doing is activating WISP-H, a hybrid histidine kinase that in turn phosphorylates Rho-R. Rho-R in turn activates BurA and BurB, which together cooperate with RPON to activate synthesis of BEP. And when we look at the electron micrographs, what we find, hopefully, you can see is that while wild type does not produce much in the way of attachment, the WISP mutations that activate via gain of function mutations in WISP-E produce these clusters that bind both by EPS and by fimbrial attachment. And those that are more fimbrial dependent but less dependent on polymer production wind up producing these tiny mucoid variants, which are activated by single direct mutations that activate WISP-H. So to conclude, RSCVs frequently and rapidly evolve when Burkholderia is selected for biofilm growth, both in vitro and in vivo, during chronic infections of the cystic fibrosis airway. These are often caused by derepressing mutations in WISP that constitutively activate Rho-R, which in turn activates BurA and BEP production. BurA and BurB together also upregulate fimbrial production that enhance aggregation. And this system then has the ability to mediate attachment by EPS and Fimbri, balancing their relative contributions, where different mutations in different parts of the systems create different attachment phenotypes. I want to thank funders and thank you for your attention.